Good morning to all my students. Still, we are under lockdown. We are still under the cage because of one small virus called coronavirus, which is so small that it doesn't belong to even a living organism, but they are just a nucleic acid covered by protein, which is an obligate parasite. And that particular coronavirus is infecting our respiratory system, the nasal cavity, the sinus area, and then throat, and then alveolar of the lungs. So it's affecting our respiratory system, and even the brain signaling, the brain part where it controls our respiratory system. So this particular virus is affecting our respiratory system. So the topic which I'm going to talk today is about something related with respiration, and the subject which I'm going to talk today is biochemistry. Biochemistry, when you understand that every forms and functions of biology we try to understand through chemical process. So this is what about biochemistry. The main hero of today's talk is glucose. Glucose is a breakdown of glucose is happening in all types of living organism. In almost all the living organism, glucose breakdown is there to generate energy. So how glucose is formed? During photosynthesis, the sunlight energy comes in the form of energy package. This energy package is used by plants to happen a reaction of carbon dioxide react with water and forming glucose in the presence of sunlight and chlorophyll. So this sunlight comes in the form of energy package, and that package, each package is converting some kind of glucose. So carbon dioxide and water forming glucose, that is what we call photosynthesis. So once this glucose is formed, this glucose has to store it, if not in use at the moment. So it has to be stored inside the cell. So the storing of glucose has to convert to some other compound. So it has to store in the form of sucrose. Sucrose is glucose plus fructose. So glucose and fructose together form a sucrose. So sucrose is a disaccharide, whereas glucose and fructose are monosaccharide. These two monosaccharides come together and form sucrose, and they store inside the plant cell. So another compound or molecule which is storing inside the plant is starch. Starch is a polymer of glucose. So many glucose units form starch, and that is again storing inside the plant cell to be used later. So sucrose and starch is stored in plants. Others I'll not talk right now. And then if we talk about animals, in animals it is stored in the form of glycogen. Glycogen is also a polymer of glucose, but it differs with starch with the branching or in the structure of the molecules. So basically they're both made up of polymers of glucose. So in animals, glucose is storing as glycogen, not only in animals, in fungi and in bacteria also. So glucose is stored in cell of plants and animals in the form of sucrose, starch and glycogen. So this is about anabolic reaction, where from carbon dioxide and water form glucose, and this glucose is formed into this sucrose, starch, and glycogen. But today's topic, we are mainly concerned with respiration. Respiration involves this glucose to break down. So break down, and then it has to form to carbon dioxide and water. So what is the fate of glucose in respiration? So glucose can go two ways, either this way or either that way. So if it goes oxidation via pentose phosphate pathway, it forms ribulose 5-phosphate. So that is one part. Another part, if it takes oxidation via glycolysis, then it converts into pyruvate. So these two parts the glucose can undertake during respiration. In our syllabus under respiration, we are going to study three things. One is glycolysis, another one is Krebs cycle, and the third one is electron transport chain. I'll take glycolysis today, and the two subsequent lecture I'll be taking up in the next two lecture. Let us see what is respiration and their different types. The phenomena of release of energy by oxidation of various organic molecules for cellular use is known as respiration. There are two types of respiration. The first one is aerobic respiration, and the second one is anaerobic respiration. Complete oxidation of organic substance in the presence of oxygen is called aerobic respiration. Whereas complete oxidation of organic substance in the absence of oxygen is known as anaerobic respiration. Let us see what is glycolysis. Glycolysis derived from two Greek words, which are glykis, which means sweet. Second one is lysis, which means splitting or breaking down. So these are the two words. So sweet and splitting. So if we want to understand about glycolysis, then we should know the word, the word which from where it derived from. It derived from two words, glykis and lysis, which means breaking down of sweet thing. So if we say in a more in a better way, then it is a degradation of a molecule of glucose. So the sweet is glucose in series of enzyme catalytic reaction to yield two molecules of 3-carbon pyruvate. Two molecules of 3-carbon pyruvate. So this glucose breaking down into 10 steps and then forming pyruvate. So that is glycolysis. So glycolysis take place inside the cytosol of the cell. When I say cytosol, cytosol is the protoplasmic part where nucleus, excluding nucleus, liquid part. So that is what we call cytosol. Glucose is a hexose ring compound polyhydroxy and functional group aldehyde. This is glucose. Glycolysis usually take place in 10 subsequent steps. These 10 steps can be again subgrouped into two. One is the preparatory phase, 1 to 5, fifth step up to till the fifth step is preparatory phase, and 6 to 10, that is the payoff phase. So in the first step, glucose is converted to glucose 6-phosphate. In this reaction, ATP is consumed. So ATP means adenosine triphosphate. ATP is an energy currency in cell. So we know currency means money. So we buy things with money. And inside the cell, the energy, that is the process are done through energy, and this energy is given by ATP, and ATP is just like a currency inside the cell, energy currency inside the cell. So ATP, adenosine triphosphate, one of the phosphate of 3 phosphate is phosphorylated and then added to glucose in the 6th carbon number and form glucose 6-phosphate. So glucose plus ATP that form glucose 6-phosphate, and also another compound ADP, adenosine diphosphate, is formed. So the enzyme involved in this reaction is hexokinase. So when we say the enzyme kinase, that means we know the ATP is involved. Where ATP phosphorylation is taking place, then we call it then the enzyme hexokinase is involved. The second step is glucose 6-phosphate converted to fructose 6-phosphate. So from converting of glucose 6-phosphate to fructose 6-phosphate, the enzyme phosphohexoisomerase is involved. This is a reversible reaction. So glucose 6-phosphate can convert to fructose 6-phosphate, and fructose 6-phosphate also can come back to glucose 6-phosphate. And so there is this type of reaction is just an isomerization. So fructose and glucose, they have the same molecular formula. The only difference is in structure. So changing of the structure, so that is glucose and fructose is an isomerase. So the enzyme which is involved also is an isomerase enzyme. So phosphohexose isomerase. And from here, fructose 6-phosphate to fructose 1,6-bisphosphate. In the next step, the step 3, it is converting fructose 6-phosphate to fructose 1,6-bisphosphate. So in the first carbon and in the last carbon, carbon number 1, 2, 3, 4, 5, 6. In the 6th carbon number 1 phosphate is attached. Carbon number 6, another phosphate is attached. So that is what we call fructose 1,6, since it is in carbon number 1 and the carbon number 6. 1,6-bis means 2 phosphate, bisphosphate, and bis means they are not in a consecutive carbon number, but in the different far away carbon number. So that's why bisphosphate. So here ATP is again involved. So ATP, from ATP, 1-phosphate is absorbed by carbon number 1 of fructose. So here fructose 1,6-phosphate is formed by addition of one more ATP. So this step number 1 and step number 3 is also known as priming reaction. So priming is taking place in these two reactions. That is why it's also known as priming reaction. The enzyme involved in this is phosphofructokinase. You know, in the first one also, when we say hexokinase, then phosphorylation is taking place. And then here, in the third step, again, kinase, we know that phosphorylation is taking place. So, fructose 6-phosphate plus ATP gives, in the presence of the enzyme phosphofructokinase, is converted into fructose 1,6-bis-phosphate. So this is also known as a priming reaction, because ATP priming is taking place here. In the formation of the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, this fructose 1,6-phosphate is broken down, is cleaved, is split into two molecules. One is glyceraldehyde 3-phosphate, another one is dihydroxyacetone phosphate. And this dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, they are also interconvertible by an enzyme known as triose phosphate isomerase. Triose phosphate isomerase. So since fructose is 6 carbon number, and these two products are 3 carbon each, so this glyceraldehyde 3-phosphate has to undergo glycolysis reaction till pyruvic acid, and this dihydroxyacetone phosphate has to convert to 3-acetyl glyceraldehyde 3-phosphate. This glyceraldehyde 3-phosphate, now it will have two compounds of three glyceraldehyde 3-phosphate. Dihydroxyacetone will also convert to glyceraldehyde 3-phosphate. So the next reaction, the reaction which is going down, it will have two molecule each. Let us see the payoff phase, that is from step 6 to step 10 is also known as payoff phase. Now step 6. In step 6, oxidation of glyceraldehyde 3-phosphate to 1,3-diphosphoglycerate. In this step, it is catalyzed by glyceraldehyde 3-phosphate dehydrogenase. And this is the first of the two energy conserving reaction of glycolysis that eventually lead to the formation of ATP. This aldehyde group of glyceraldehyde 3-phosphate is dehydrogenated not at a free carboxyl group but to the carboxylic anhydride with phosphoric acid. This is called acyl phosphate. The acceptor of hydrogen is in the glyceraldehyde 3-phosphate dehydrogenase reaction is NAD positive. This will reduce to coenzyme NADH. In step 7, 1,3-bisphosphoglycerate is converted to phosphoglycerate. Phosphoryl transfer from 1,3-bis-phosphoglycerate to ATP. The enzyme phosphoglycerate kinase transfer the high energy group from the carboxyl group of 1,3-bis-phosphoglycerate to ATP, forming ATP and 3-phosphoglycerate. So these step 6 and step 7 are energy coupling process. Step 8. This is the conversion of 3-phosphoglycerate to 2-phosphoglycerate. So this conversion of phosphoryl from carbon number 3 to carbon number 2 is because this reaction is because of the presence of the enzyme mutase, phosphoglycerate mutase. So this phosphoglycerate mutase allow the movement of phosphoryl from carbon number 3 to carbon number 4. And in this reaction, magnesium ion is present. Step 9 is the dehydration of 2-phosphoglycerate to phosphoenol pyruvate. The enzyme involved here is enolase, and it is a reversible reaction. The last step, that is the 10th step, it is the conversion of phosphoenol pyruvate to pyruvate. Here in the process, this ATP reacts with phosphoenol pyruvate and gives a ATP, adenosine triphosphate, and then pyruvate. It is done by the enzyme pyruvate kinase. Kinase is an enzyme which is responsible for production of ATP. And this is the second ATP forming reaction, and this is a substrate level phosphorylation. So the step 10 is the last step of glycolysis. So let us see the fate of pyruvate. So from glucose to converting to pyruvate is glycolysis. Now after this glucose become pyruvate, let us see the fate of pyruvate, where this pyruvate goes. So under anaerobic condition, this pyruvate will undergo this fermentation, and it will convert this pyruvate into 2-ethanol plus carbon dioxide. So fermentation, one pathway is fermentation, and another part which will be in that is also in anaerobic condition. So that is formation of lactate. That is usually happen in our muscle. Fermentation to lactate in vigorous contracting muscle in erythrocyte, in some other cells, and in some microorganism. So lactate formation will be there. So under aerobic condition, it will this pyruvate will convert into 2-carbon compound, that is 2-acetyl coenzyme A, and then this 2-acetyl coenzyme A will undergo Krebs cycle, and after that carbon dioxide and water will be given out. This happens in both animals, plants, and microbial cells undergo aerobic condition. ATP formation coupled to glycolysis. The equation of glycolysis resolved into two processes, exergonic and endergonic. The conversion of glucose to pyruvate. So this glucose plus 2 NAD gives 2 pyruvate plus 2 NADH plus hydrogen ion, 2 hydrogen ion. This is exergonic reaction. And another reaction is the formation of ATP from ATP and inorganic phosphate. 2ADP plus 2P gives 2ATP plus water. The overall reaction of the equation of glycolysis is given as glucose plus 2NAD ion plus 2ATP plus inorganic phosphate gives 2-pyruvate, 2NADH plus hydrogen ion, 2 hydrogen ion plus 2 ATP plus water, 2 water. So let us see the fate of pyruvate. So from glucose to converting to pyruvate is glycolysis. Now after this glucose become pyruvate, let us see the fate of pyruvate, where this pyruvate goes. So under anaerobic condition, this pyruvate will undergo this fermentation, and it will convert this pyruvate into 2-ethanol plus carbon dioxide. So fermentation, one pathway is fermentation, and another part which will be in that is also in anaerobic condition. So that is formation of lactate that is usually happen in our muscle. Fermentation to lactate in vigorous contracting muscle in erythrocyte, in some other cells, and in some microorganism. So lactate formation will be there. So under aerobic condition, it will this pyruvate will convert into 2-carbon compound, that is 2-acetyl coenzyme A, and then this 2-acetyl coenzyme A will undergo Krebs cycle, and after that carbon dioxide and water will be given out. This happens in both animals, plants, and microbial cells undergo aerobic condition. The overall balance sheet shows a net gain of ATP. The left hand side of the following equation shows all the inputs of ATP, NAD, ADP, and inorganic phosphate, and the right hand side shows all the output. Keep in mind that each molecule of glucose yields 2 molecules of pyruvate. Glucose plus 2 ATP plus 2 NAD positive plus 4 ATP plus inorganic phosphate give 2 pyruvate plus 2 ATP plus 2 NADH plus 2 hydrogen ion plus 4 ATP plus 2 water. Cancelling out common terms on both sides of the equation give the overall equation for glycolysis under aerobic condition. Glucose plus NAD plus 2 ATP plus inorganic phosphate give 2 pyruvate plus 2 NADH plus 2 hydrogen ion plus 2 ATP plus H2O. So the net equation gives 2 ATP. Let me now summarize glycolysis. In glycolysis, glucose is broken down into pyruvate. It undergoes 10 enzymatic reaction. The first 5 enzymatic reaction is known as preparatory phase. In this preparatory phase, ATP priming takes place, and in this reaction, cleavage of glucose into 2 compound, that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate reaction take place. The second part is the payoff phase. Here substrate level phosphorylation take place. NADH and ATP is formed. In the second phase, the product at the final step, the product pyruvate is formed. So this is all about glycolysis. Thank you for keenly listening. We'll see you in the next lecture.